In this lecture, we will continue the discussion of the hypothalamo-pituitary thyroid axis. It is a continuation of the lecture on the HPT axis posted on the website. In the first half of this lecture, we basically looked at the gland 1, hormone 1, and gland 2, hormone 2 to some degree. Now we are focusing on the gland 3, hormone 3. And as you recall, I'll go to the computer now. Get my pip on here. We are discussing the hypothalamo-pituitary thyroid axis, and we had talked about the TRH neurons and its regulation through negative feedback, through the links on the website to endotext and the ACNP text. There was information on the endocrine transduction system. Remember, we talked about that, the neurotransmitter effects on this, converting nerve impulse to neuroendocrine information. So the TRH discussed there, as well as a little bit about the negative feedback regulation on that. We talked about the thyrotropes, using lactotropes as another example for calcium-mediated release. Talked about second messenger systems, a little bit about the receptor, and the TSH molecule itself, the two gene components, a little bit about the promoter region with reference to the website for more on that. And now we're focusing on the thyroid gland, looking at the thyroid hormones, and then I think we take a bit at the end to go back and reflect on negative feedback at the level of the anterior pituitary. So we had left with this slide at the previous lecture, focusing on the thyroid gland itself. We looked at the follicles that contain the thyroglobulin and the follicular cells that surround them. Now remember, these are spherical type of structures, so picture a basketball filled with a bunch of jello, I guess would be one way of thinking about it, a thick basketball lined with the cells. So it's a three-dimensional follicle or spherical structure. Of course, when you cut it and look on the cut edge, it's roughly circular, right? So you can see that some have more colloid than others, and so on and so forth. And that can simply be from sections cut through different levels. Picture cutting through the basketball. So if you had a section through this follicle right here and looked at it from that direction, it might look this diameter. So it's hard to say in just looking at one section whether any are bigger than the other. This might be cut obliquely. This might be a nice cut right through the center of one, something like that, and this at the tip. So it's rather hard to say. But certainly this scalloping at the edge is evidence that there's some type of marginal activity going on where the follicular cells are interacting with the colloidal cells. We had imagined ourselves riding a TSH molecule coming from a capillary diffusing out down our concentration gradient into the interstitial fluid compartment, bouncing around, bumping into receptors. Remember, this is constantly ongoing with TSH being degraded in the interstitial fluid compartment and fresh TSH coming through to sustain some certain interstitial fluid concentration of TSH such that there is a sustained level or a grossly increased level or overall increased level of TSH receptor stimulation on the basal membrane of the follicular cells, altering intracellular events such that the apical membrane interacts with the colloid to contribute to the formation of the thyroid hormones. So picture that when you're looking at just the nucleus here with some cytoplasm surrounding it of a follicle cell. We'll see an illustration in a moment that is very illustrative of a follicular cell. So the thyroid hormones, they're derivatives of the amino acid tyrosine. Remember early on we talked about tyrosine being a precursor to be converted into the catecholamines and as well to the thyroid hormones, and now we'll take a closer look at it. It is bound covalently to iodine, So we know that iodine is a critical component in the diet. The two principal thyroid hormones are thyroxine, known affectionately as T4 or L3,5,5' tetraiodothyronine, and triiodothyronine, the H is missing there, T3 or L3,5,3' triiodothyronine. We see the molecular structures here on the bottom. The tyrosine molecule itself, you see that there's just one phenol type ring or benzene ring, whatever term you want to use for it, aromatic ring. And in the thyroid hormones, there are two of these. So obviously, two tyrosine amino acid residues are consumed to make one molecule of a thyroid hormone, whichever one is made, T4, T3, or the reverse T3. And we'll discuss these in a bit more detail in a moment. Notice that the carboxy and amino terminal remain only on one side of this molecule, so it is an asymmetrical molecule. I have to be honest, I don't know which portions of the molecule are critical or key for binding to the receptor. I am assuming that this arrangement of the three iodine molecules, or elements on here, is critical because when reverse T3, where we see there's two, let's say, distal to the amino terminal end of this structure, and one proximal, this is non-functional. However, it's described as a dominant negative molecule in that it can bind to the receptor, probably doesn't have as high of an affinity, but it does not activate the thyroid receptor. Had to struggle to pull out that word dominant negative there, didn't I? Okay, let's move on. So a large majority of the thyroid hormone secreted from the thyroid gland is T4, but T3 is the considerably more active hormone. We already saw the deiodinase, I think I pronounced it correctly, that converts, is an intracellular enzyme within target cells that converts T4 to T3. So some T3 is secreted. Most of it is derived by the deiodination of the T4 in the target cells, of the target tissue, right, of the target glands, especially liver and kidney. And the deiodination of the T4 also yields that reverse T3, a molecule with no known metabolic activity. So it's not really understood why it is produced. It may just be one of those enzymatic events that can occur randomly, and it may not contribute any functional benefit to the organism, acting as a dominant negative molecule, in essence then almost an antagonist to the thyroid hormone receptor. So not much is known about that other than a few of its properties and the fact that it does occur. The thyroid hormones are poorly soluble in water, so they are more lipophilic than they are hydrophilic. There are actually specific transporters to get them across the cell membrane, and you can seek out more details on that in your own reading. I think I mistakenly said that they were freely diffusible through the membrane, and in essence the dynamics would be very similar, but I believe that there is a carrier molecule for that. Anyway, more than 99% of each of these circulating in the blood is bound to carrier proteins. So classically, when the thyroid hormones are measured in the plasma, the free versus bound ratios are calculated. Just a moment, I'm seeing strange things on my monitor here. I don't have any user controls. I hope you're recording this in the back there. The principal carrier of the thyroid hormones is the thyroxine binding globulin, and it is a glycoprotein. It is synthesized in the liver, And there are two other carriers of importance are the transthyretin and the albumin. The carrier proteins allow the maintenance of a stable pool of thyroid hormones from which the active free hormones are released for uptake by target cells. So basically what's being described here is there's a dynamic process of being bound. Remember, this is not covalent binding. It is typically hydrogen hydrogen bonding, holding the hormone at the carrier molecule for a brief period of time. So that's the dynamic binding and unbinding with the transport proteins. So at any one point in time, there's a certain amount in any one area as blood is passing through a capillary. There's a certain amount that is unbound that can diffuse out. So that's the basic idea of what's occurring there from the physical properties. I'm just going to go check, make sure they're capturing all this in the recording. So, hello, I'm back again. Just a little strange thing here with my controller, but I checked in the back room, they're recording this, so you are seeing what I'm pointing at in addition to hearing what I'm saying. So let's continue on then. Let's now discuss the synthesis of and the secretion of the thyroid hormones. So the entire process occurs in three major steps. So remember, we've got this thyroglobulin that obviously is acted on in some way and broken down to produce the fully active, biologically active hormone that is secreted. So let's keep that in mind as we proceed through this discussion. So there's at least in some ways these are analogous to those used in the manufacture of integrated circuits. So I've given you my source for this information that I have in presenting these slides. It's on the last slide of this presentation, so I'm not taking credit for this idea of comparing it to integrated circuits. I just thought it was very interesting and left it in place. So the production and accumulation of the raw materials, so in the case of the ICs, a large wafer of doped silicone. So obviously this is the thyroglobulin. Fabrication or synthesis of the hormones on a backbone or scaffold of precursor, like etching several ICs on the silicone wafer. So there's a lot of tyrosine residues scattered within or throughout the thyroglobulin. The release of the free hormones from the scaffold and secretion into the blood, so cutting the individual ICs out of the large wafer and then distributing them. So that seemed to be a model that might be a bit strange to most of us biologists, so we probably know a little bit more about IC production than we do using that as a model to understand how thyroid hormones are produced. So the recipe for making thyroid hormones calls for the two principal raw materials, the tyrosines, and they're, of course, in this large glycoprotein scaffolding. They're actually within it as amino acid residues in the amino acid sequence of thyroglobulin. It's synthesized by the thyroid follicular epithelial cells and secreted into the lumen at the apical membrane of the follicle colloid, of the follicle into the core of the follicle inside that basketball that we pictured, producing the jello inside the basketball. And the colloid is essentially a pool of thyroglobulin. A molecule of thyroglobulin contains 134 tyrosines, although only a handful are actually used to synthesize T3 and T4. There's just a large number of them in this protein that we would assume was evolutionarily exploited to produce this hormone. So the iodine, or more accurately iodide, the ionic form, I negative, is avidly taken up from the blood by the thyroid epithelial cells. which have on their outer plasma membrane a sodium iodide symporter. So sodium influx draws the iodide in with it, or also called the iodine trap. So once inside the cell, the iodide is transported to the lumen of the follicle along with the thyroglobulin. So now you have to picture the basal membrane and the apical membrane of the thyroid follicle cell. We have this symporter on both sides, bringing sodium in from the interstitial space, and then the iodide is then pushed out in on the apical membrane to be with the thyroglobulin. It's conducted by the enzyme thyroid peroxidase. Ultimately, which is an integral membrane protein in the apical colloidal facing plasma membrane. I'm sorry, I was wrong. I said that the symporter was on both basal and apical. It's only on the basal because sodium is higher concentration outside, so you get the concentration gradient driving it in with that sodium iodide symporter on the basal membrane. So this is actually an asymmetrical distribution of membrane proteins for the movement of iodide in at the basal and then out at the apical membrane. It's a thyroid peroxidase integral membrane protein in the apical or the colloidal facing plasma membrane of the thyroid follicular epithelial cells. This catalyzes two sequential reactions: iodination of the tyrosines on the thyroglobulin, so the iodide has to be present to do that, also known as organification of the iodide, adding it to an organic molecule, and then the synthesis of the thyroxine or triiodothyronine from two iodotyrosines. So we'll follow through with that description using this illustration. So here we see the thyroglobulin, and the two tyrosine residues that are involved are not right next to each other. They're somewhere distant from each other, such that the three-dimensional folding of the protein brings them in proximity, and the peroxidase can act on it, and the iodide is there. So the peroxidase puts two on each of them, and then continues the reaction to pull one away from its carbon that we see here, the carbon side chain, and it forms this, is that an esterification using the oxygen in there? Anyway, I'm not going to get into organic chemistry that much, huh? So through the action of the thyroid peroxidase, the hormones accumulate in the colloid on the surface of the thyroid epithelial cells. Remember that hormone is still tied up in the molecule of thyroglobulin. The task remaining is to liberate it from the scaffold and secrete the free hormone into the blood. So they're excised from the thyroglobulin scaffolding by digestion in lysosomes of the thyroid epithelial cells. So here we can see in this illustration, I know you can't see it all that well, but hopefully on your printout you can, or at the website, the source of this. The iodinated thyroglobulin is shown here with these red and green positions at the in the colloid at the margin with the apical membrane of the thyroid follicular epithelial cells. Through a process of endocytosis, that's thyroglobulin, including the iodinated tyrosine residues, as we saw, are endocytosed. You can see this is a clathrin-coated pit, meaning that this endocytotic process is going to deliver this endosome to fuse with a primary lysosome and become a secondary lysosome, and it's in the secondary lysosome that you have the enzymes that perform the cleavage, and as soon as the free thyroid hormones are released, they are diffusible in such fashion that they can diffuse out of the follicular cells at the basal membrane and through the interstitial space to the blood. Remember that the lipid-soluble hormones are not stored in vesicles and then released through exocytosis. As soon as they're synthesized, they're released. So the final act in thyroid hormone synthesis proceeds in the following steps. The thyroid epithelial cells ingest the colloid by endocytosis from the apical borders. That colloid contains the thyroglobulin with the thyroid hormone, the iodination sites. The endosomes that have that colloid fuse with the primary lysosomes containing the hydrolytic enzymes, digest it further, liberating the free thyroid hormones, which diffuse out of the lysosomes through the plasma, basoplasma membrane, interstitial space in the blood, and then when they are bound by their carrier proteins, they are taken out of the concentration gradient calculation, if you want to think of it that way. They're no longer as a single molecule in the plasma. When they are bound, they're taken out of the formula, so it sustains a concentration gradient, drawing this hormone from the cell into the plasma, out of the interstitial fluid into the plasma. So again, this is the basic concept that we put forth originally about the endocrine organs having a rich capillary bed with a significant blood flow to keep washing away the hormone that is secreted and sustaining that concentration gradient so that it's low in the plasma and high in the interstitial fluid compartment. And in the case of these molecules, it's thereby creating a draw out of the cell of these freely diffusible molecules. It's still thought that they diffuse out, as you noted in that prior slide. So now we can talk about the thyroid hormone receptors. So thyroid hormone is being carried in the plasma, binding and unbinding to the carrier molecules, and are distributed through the plasma. Remember as well, it's venous return to the heart, then the systemic arterial circulation being distributed and diffusing into the interstitial space throughout the body, sustaining some level of concentration that will be increased or decreased dynamically over time, and the thyroid hormone just randomly bouncing around and bumping into its receptor, now diffusing, being transported into the cell, target cells, and diffusing around, bumping around, and randomly colliding with the receptors in the nucleus. Okay. So the receptors for the thyroid hormone, we got there somehow, are members of a large family of nuclear receptors, as we recall. They function as hormone-activated or ligand-dependent transcription factors and are active by targeting promoter regions on target genes, as we know. In contrast to the steroid hormone receptors, thyroid hormone receptors bind DNA in the absence of the hormone, usually leading to transcriptional repression. So we talked about this as a generic type of a thing when we talked about the steroid receptor family in general. And now we see it in a specific example here for the thyroid hormone receptor, and we'll continue to consider that concept for this particular receptor. So the hormone binding is associated with a conformational change in the receptor, as you recall, causes it to function as a transcriptional factor. So ligation, conformational change, DNA binding domain is uncovered, and it now, bumping around, can bind to the DNA. Or if it's sitting there acting as a repressor with the low affinity, when it is ligated, changes conformation, higher affinity, and then it can have another effect. Typically it will be an activation. So the mammalian thyroid hormone receptors are encoded by two genes designated as alpha and beta. So two isoforms coming from two different genes. The primary transcript for each gene can be alternatively spliced. So now we have a larger number of isoforms possible, generating different alpha and beta receptor isoforms. So currently there's 4 different isoforms of this receptor: alpha 1, alpha 2, beta 1, and beta 2. Don't confuse these with the adrenergic receptors, just a similar type of approach for nomenclature. So these thyroid hormone receptors have the 3 functional domains. There's the transactivation domain at the amino terminus, interacts with other transcription factors, forms complexes, that repress or activate transcription. Considerable divergence in the sequence of this domain between the alpha and the beta isoforms and between the two beta isoforms of the receptor. A DNA binding domain, particular sequences, the TRE that we'll talk about in a moment, found, of course, on the promoter regions of target genes. A ligand binding and dimerization domain at the carboxy terminus. So as depicted in the figure below, the DNA binding domains of the different receptor isoforms are very small. So the DNA binding domain is here in the middle, kind of at the, remember this hinge region, although the term isn't being used here. Remember that there was a hinge region somewhere near here in this model of receptor. There's divergence among the transactivation and ligand binding domains. Here is the ligand binding and dimerization, the carboxy terminus, and the transactivation domain of the amino terminus, and it's, of course, alpha 1, alpha 2, beta 1, and beta 2. So most notably, the alpha 2 isoform, this one here, the second blue one, it has a unique carboxy terminus and does not bind triiodothyronine. So the different forms of the thyroid receptors have patterns of expression that vary by tissue, so tissue-specific expression and developmental stage-specific gene expression. So for example, almost all tissues express alpha 1, alpha 2, and beta 1 isoforms, but beta 2 is synthesized almost exclusively in the hypothalamus, anterior pituitary, and the developing ear. The receptor alpha 1 is the first isoform expressed in the conceptus, and there is a profound increase in expression of the beta receptors in the brain shortly after birth. Interestingly, the beta receptor preferentially activates expression from several genes known to be important in brain development, for example, the myelin basic protein, and upregulation of this particular receptor thus may be critical to the well-known effects of the thyroid hormones on development of the fetal and the neonatal brain. Of course, any place you see these underlined blue bold words in the text at the original site, there's links to other information. So the presence of multiple forms of the thyroid hormone receptor with tissue and stage-dependent differences in their expression suggests an extraordinary level of complexity in the physiological effects of thyroid hormone. Now let's consider the interaction of thyroid hormone receptors with the DNA. So the thyroid hormone receptors bind to short repeated sequences of DNA called the thyroid or T3 response elements, or the TREs, as we have indicated previously, a type of hormone response element. So a TRE is composed of two AGGTCA half sites separated by 4 nucleotides. So AGGTCA. So are you beginning to store into your memory all these different consensus sequences for the response elements that have been assigned? The half sites of a TRE can be arranged as direct repeats. The palindromes, this is the term that I've been searching for all semester, and I just couldn't come up with, which are, of course, are the inverted repeats. So it would be AGGTCA, and then with 4 in between, then would go ACTGGA. And that's the palindrome, or the inverted repeats. Some can be direct repeats, and others would be the palindromes, or the inverted repeats. So the DNA binding domain of the receptor contains 2 sets of 4 cysteine residues, and each set chelates a zinc ion, forming loops known as zinc fingers. So this is in the family, this transcription factor is in the family of zinc finger domain transcription factors, or DNA binding proteins. A part of the first zinc finger interacts directly with nucleotides in the major groove of TRE DNA, while the residues in the second finger interact with the nucleotides in the minor groove of the TRE. Hopefully you recall that the DNA helix, the helical formation has a major groove and a minor groove. Excuse me. Thus, the zinc fingers mediate specificity in the binding of the TREs. Create that opportunity to get in there and have access to these sequences of nucleotides such that you can have stoichiometry appropriate for specificity and selectivity. The thyroid hormone receptors can bind to TRE as monomers, as homodimers, or as heterodimers with the retinoid X receptor or RXR. We see these illustrations here. This concept was introduced early on when we talked about steroid and thyroid hormone receptors in general. The RXT is another member of the nuclear receptor superfamily that binds 9-cis-retinoic acid, and the heterodimer affords the highest affinity binding and is thought to represent the major functional form of the thyroid receptor. So here we see the monomer binding to a half TRE, although we see that this is a direct repeat. It's not the palindromic type. A monomer of alpha, or it could be beta. The homodimers can be alpha-alpha, alpha-beta, or beta-beta, still considered as homodimers. And then the heterodimers, alpha with the RXR or beta with the RXR, binding to the AGG-TCA, and then the AGG-TCA, so this is a direct repeat in this case. In this instance, they're all shown to be ligated with their cognate ligands. So the thyroid hormone receptors bind to TRE DNA regardless of whether they are occupied by T3. The biological effects of TRE binding to the unoccupied versus the occupied receptor are dramatically different, and we'll look at some specific details of this to help understand, exemplify how these transcription factors impact DNA and have effects on increasing or decreasing the statistical likelihood or frequency with which RNA polymerase might be able to bind to the initiation site, and thereby impacting expression rate. In general, binding to the thyroid hormone receptor alone to DNA leads to repression of transcription, whereas binding to the thyroid hormone receptor complex activates transcription, as we had said before. Now let's consider this in detail to help understand the mechanism by which this occurs. In the ligand-free state, or the unligated state, we see the co-repressor complex shown here, the TR, thyroid hormone receptor, and the RXR receptor shown here, sitting on the DNA in the unligated form, complexed with some other molecules. The transactivation domain of the T3 free receptor, excuse me, try that one more time. The T3 free receptor, as a heterodimer with RXR, assumes a conformation that promotes interaction with a group of transcriptional co-repressor molecules. A part of this co-repressor complex has histone deacetylase activity, or HDA. We see that here. So it is the HDA is associated with the formation of a compact, turned off conformation of the chromatin. So it tightens the coiling and decreases the availability, or the amount of space available for RNA polymerase to randomly bump into its site for initiating transcription. So again, a lot of it has to do with the tightening or loosening of all of the coiling, all the way up to the super coiling, or whatever the terms are, for the DNA. So the net effect of recruiting these types of transcription factors is to repress transcription from affected genes, target genes. So in the unligated form, it is repressing the target genes, It may not be turning them off completely. It may just be subduing or sustaining a baseline level of expression. So keep in mind, in the unligated form, probably a low affinity for the DNA and a high affinity for this compressor complex. Once ligated, that affinity likely is going to change. It's definitely going to change for this co-repressor complex in that the affinity will decrease, so these proteins will unbind and move out of the way. And I don't know the exactness of that. However, these are predictions that we can make in our general understanding that we hopefully have improved through this course of how biological molecules interact, especially protein interactions. The ligand binding, ligand bound state, or the ligated state. So we have Rx3 and just in this case the T3 on the TR receptor. So the binding of the T3 to its receptor, conformational change, makes it incompetent to bind the compressor complex. So it has a lower affinity, right? But the competent or increases its affinity to bind another group of proteins, coactivator proteins. So here we see the coactivator protein complex, and we already see this new molecule there that is an HAT. So it puts on its hat when it's ligated. The coactivator complex contains histone transacetylase or acetylase activity, and this imposes an opening configuration in adjacent chromatin. The coactivator complex associated with the T3 bound receptor functions to activate transcription from the linked genes. There are exceptions to this rule. A growing number of specific proteins have been identified in members of the co-repressor and coactivator complexes that are described. It should also be mentioned that there are several exceptions to the scheme described above. As mentioned, the alpha-2 receptor is unable to bind T3 and acts similarly to a dominant negative mutant of the receptor. So here we see this term again. But its carboxy terminus can be differentially phosphorylated. Remember our discussion of phosphorylation of the receptors, transcription factors, which affects the DNA binding and dimerization. So also the beta-2 isoform apparently fails to function as a repressor in the absence of T3. And here at the bottom of this slide is the link to the source of this information. And this ends our discussion of the hypothalamo-pituitary thyroid axis. I appreciate your time and attention and your tolerance or toleration, I guess, of my pronunciation blunders along the way. We'll see you again soon. Bye-bye.